cancers are at root a very simple disease, which is they're caused, as far as we can tell, by mutations that occur in certain cells. And in Burkitt lymphoma, these mutations occur in the antibody-producing cells, the so-called B cells or B lymphocytes. Now, the interesting thing about Burkitt lymphoma that we've known for some time is that they all share a particular type of mutation that affects a gene called MYC. And MYC is somehow involved in driving the replication of cells. Not just the replication of cells, but all the things that cells need when they replicate, because they replicate in the body and they need to talk to other cells. And MYC regulates that by regulating the genes that are expressed or suppressed when MYC is activated. And we know that all Burkitt lymphomas have activation of this gene MYC. That is, it's switched on in a way that it is not normally switched on, in a way that it can't be turned off and at levels that are much higher than in normal cells. And there is, I think, no shadow of doubt that the activation of this MYC oncogene is the platform upon which all Burkitt lymphomas reside. But not all Burkitt lymphomas are the same, and not all the patients who get Burkitt lymphoma are the same. And the reason for that is that MYC provides the platform, but then mutations arise randomly during the evolution of these cancers in each individual patient. And indeed, even within a patient, there will be diversity of mutations and different, if you like, species of the tumor that grow out all of the time. And what we need to do is find out exactly what those mutations are, because they add in necessary ways, we believe, to the survival of that tumor and therefore lead to the death of that patient. And so what we really need to do now is take these tumors apart at the sequence level, the code that drives them, and find out where the errors are. And to do that, we need to basically sequence the code of tumors, Burkitt lymphomas, from individual patients, and try and find out how similar they are and how different they are, and how the similarities can lead us to thinking about drugs that may work generally against the disease, and how the differences will enable us to treat patients who otherwise would be very difficult to treat.